So we talked about several types of databases, and there is another way to classify these databases, that can be primary versus secondary biological databases. So what are primary databases? The primary databases contain the information derived from the data which is derived from the research in the laboratory, meaning it is experimentally derived data, like the nucleotide sequence which we represent in primary database. It comes from the result of experimental sequence determination. So the protein sequence or the macromolecular structure, it is directly coming as a result of research. And the whatever databases we discussed earlier, most of them were primary databases. But the databases can be secondary databases also, and the secondary databases comprise the data derived from the results of analyzing the primary data. Like I tell you again, I think we first define what is secondary database. Secondary databases comprise data derived from the results of analyzing primary data. They are highly curated. Compared to primary databases, they are highly curated, often use a complex combination of computational algorithms and manual analysis, meaning manual analysis apart from algorithms is equally important here. And with these two methods, meaning computational algorithms and manual analysis, we interpret the data and we derive new knowledge from the publicly available records of science. There have been several examples of deriving this knowledge, but the important thing here is that they are mainly based on the information stored in the primary data. They are simple archives of the translated sequence data from identified DNA sequences. So what we are trying to tell you here is that they can be very simple, like they would be just telling you the open reading frames in the DNA sequences, but they can be very advanced also, like they might have additional annotation and information related to the higher levels, like levels of information regarding the structure or the function of a protein. So they can be very advanced also. So secondary database, they again it contains the information derived from primary sequence data, but the information derived is in the form of regular expressions, and these regular expressions, we talk of three regular expressions here. It may be a pattern, it may be fingerprints, or it may be profiles or blocks. So the main theme on which this secondary secondary databases are based on the multiple sequence alignments, and we deduce the conserved regions. Which are the conserved regions? They show very little or no variation in the structure across the constituent sequences, and these conserved regions, they are called as motifs. So motifs is the mainstay of development of these secondary databases, and motifs are the conserved region which show no variation across the different sequences. And in the last presentation I showed you one of my multiple sequence alignments where there were several conserved regions which were same in different sequences. Similarly, the motifs are very important because they indicate the vital biological role in that sequence, meaning these are the regions which are carrying the function of that molecule, and they are very crucial to the structure and function of protein, meaning in the protein folding they have very important role, in the protein binding. So they basically determine the intramolecular interaction, and that in turn determines the structure and function of the protein. So if we concentrate on motifs, we can find out the common conserved regions in the sequences and study the functional and evolutionary details in the organisms. So motifs are very important in determining, and there are three different like I have just in this picture before going to the three different types of representation of motifs, I thought I just show you the pictorially how motifs are represented. So here in the red color you see the motif of a family, and if you see the yellow color, that is the motif for a subfamily, meaning you see this region same across the families of the proteins. Next is the first secondary database example, that is the ProCyte database. It was the first secondary database which was developed, and the protein families based on most conserved motifs are found, where the ProCyte are found, and within the ProCyte motifs are encoded as regular expression, and this is called as pattern. So ProCyte database was the first one to be developed, and it of course it depends on the motifs, but we find out the regular pattern, regular expression of these conserved motifs. So what we do is we do multiple sequence alignment, I told you earlier, and we also do the manual inspection. So software based multiple sequence alignment is fine, but manual inspection is equally important here, is that we manually look at the alignment. So this is how the ProCyte database is developed, that we see the pattern in the motifs, and what information we get out of this is the protein domains, families and functional sites. We can derive this information from this ProCyte database. Here is the web view of this database, and you get two files. One gives the pattern and list of all matches of the pattern, whereas the second one gives you the details of family, description of biological role, etc. So this is based on the primary database, but the information is derived on the basis of pattern in the motifs. The next example for secondary database is PRINTS, and PRINTS is a collection of diagnostic protein fingerprints. So PRINTS is what? PRINTS is a collection of fingerprints, and fingerprints are diagnostic in nature, meaning we can identify the protein family with fingerprint. So how do we define fingerprint? Fingerprint is a group of conserved motifs that is used to characterize a protein family. Fingerprints can encode protein folds and functionalities more flexibly and powerfully than can single motifs. Full diagnostic potency deriving from mutual context provided by motif neighbors. So fingerprints are more powerful than motifs, and ProCyte and PRINTS are both manually annotated secondary databases. So they do depend on multiple sequence alignment, but manual curation also is quite important in these two databases. So again, coming back, I was just trying to tell you the motifs. The fingerprints is what happens is the fingerprint can be said to be a family motif. So the motifs or the patterns in the motifs, when we have them, we consider few of them in a particular pattern, and that pattern can be used to identify a protein family. We call that as a fingerprint. So usually the motifs do not overlap, but they are separated by a sequence, and it may not be looking continuous to you here in this extended proteins in amino acid sequence, but when they occupy their three-dimensional structure in this space, they usually lie very close to each other. So, but it was just to present once again that fingerprint is the group of motifs. So, the PRINTS database web view once again. The third one here in the secondary databases is the block database. So, the motifs here are called blocks, and they are generated automatically by highlighting and detecting the most conserved regions of each family of proteins. So, it is automatically created by in the different families, and we know that in this family this is the most conserved region that we refer to as blocks. The good thing or the new thing here is it is fully automated. We don't do any manual curation here. We do what? We use keywords and sequence searching are the two important features of this database, and blocks are ungapped multiple sequence alignments representing conserved protein regions. So, if you like to define what are blocks, they are ungapped multiple sequence alignments and they represent the conserved protein regions. Going further to the last type of secondary database, that is profile database. Profile database again is used to find out the most conserved regions in the sequence alignment. But the new thing here is that we have indels used here, meaning indels is insertion and deletions. In the multiple sequence alignment, we can insert some bases, we can delete some bases to make it a perfect match, and these insertions and deletions are weighed according to the matrices. So, the new thing here was these indels, and they are allowed, and we can predict the distant relationships among the sequences, meaning the changes also is possible to predict here when we are trying to change the sequence by insertion of a gap or deletion of a gap this way. So, that was about the secondary database basis. I cited few examples, 4 examples of the secondary databases, and what we understand out of all these examples is that we have the annotation and information regarding the structures and functions within this secondary database, and they basically require advanced computational knowledge, algorithms, and also require manual curation. So, coming to the last segment of the present talk, that was data retrieval systems. So, what are data retrieval systems? The data retrieval means obtaining the data from a database management system such as DBMS, and the data is represented in structured way and there is no ambiguity. So, that makes it the most important, that it should be searchable and it should be presented in a structured way, and there is no ambiguity in the data. So, also what we do is that we have our set criteria for search, meaning we have to have a query to retrieve the data, and then what happens is the DBMS, that is the software for managing these databases, this selects our query and then this retrieves the data which is stored in the database in a file, or maybe it may be printed or viewed on the screen. So, if we must also know before querying that there is a query language, that is SQL, structured query language. It is used to prepare queries, and the tables and the databases can very quickly be searched through this query language SQL. Here is an example which I specifically picked because I wanted to explain that we should know what happens when we make a query on the surface. So, this is an interface which I took from any web view, and I am sure each one of us are very familiar with the forms or maybe BLAST here. Suppose I just want to tell you what steps happen when we make a query. So, this is the meaning the database system DBMS or the querying data retrieval. We can divide this into 2 parts. One is the front end which we see on the screen, and the other one is the back end where our data is stored. So, if we divide the sequence of events which happens if we make a query, suppose like in this BLAST, if you insert a query, a sequence here, what happens is in the front end this form is made by HTML or DHTML, then we have few front end languages like JavaScript or maybe VBScript. What it will do? Suppose you have added a DNA sequence and you are going to search it as DNA, how it will make sure that this should not have any M or P maybe or S, because then it is not a DNA sequence. So, if you add any M here, it will give you an error. That all is a function of front end languages, meaning same way for email, if you are writing an email in a form and if you see if you didn't add the ampersand, a sign here, what will happen? It will reject your request. So there is one checking at the front end, and that is done by VB script and JavaScript. Once this VB script and JavaScript says that your information is okay, it will take it to the back end, and there is one connecting languages like maybe CGI Perl or ASP. What they do is they take your request and convert it to structured query language, which I told you just now. So what ASP and CGI Perl or the backend language is doing, they are taking up your query, converting it to SQL. So suppose you have searched for a sequence, what it will do is it will take your sequence and it will, by the help of SQL command, it will search across all the tables and retrieve the information. But the information again is SQL information that goes back to these CGI Perl or backend languages, and these ASP or CGI Perl converts it back to the HTML, and then they reach to your screen as a web view once again, or they might deliver you as a file. So this all happens when we are trying to retrieve a data, and understanding of these steps really enhance our understanding and knowledge how it is functioning. So in the data retrieval system, what is it doing? It allows us to search across multiple molecular biology databases. So it provides links to the relevant information and entries that match the search criteria. We have three systems that differ in the databases: the search and the links they have to other information. The first one is SRS, that is sequence retrieval system. The second is entries, and third is dbget. So let's look at them one by one. So SRS is sequence retrieval system, is a homogeneous interface which covers 80 biological databases. You can retrieve data from 80 biological databases at one place. This is developed by European Bioinformatics Institute, EBI, and includes a lot of databases like metabolic pathway, transcription factor, BLAST, FASTA, protein 3D structures, genome mapping, mutations. So it includes, but it's a very popular one, and it covers up to 80 biological databases. So this is just a web view to show you that this is how it looks. So you can make your query, and after you enter your query, you got to choose the alignment algorithm like maybe cluster W or phylab, whichever you want to enter, and it will give you the result. But SRS is quite popular and is highly recommended for use. Then we also have entries, that is developed by NCBI. Entries is very easy to use, but it offers limited information to search. Entries covers over 20 databases, including the complete protein sequence, data from PIR International or maybe SwissProt, PDB, nucleotide sequence data from GenBank, and includes information from EMBL and DDBJ also. This is a web view for entries NCBI. Then the third type here is dbget. What happens in dbget is an integrated database retrieval system developed at the University of Tokyo. This also provides access to 20 databases, one at a time, and having more limited options, dbget is less recommended than the other two. So summarizing, what did we understand or about these databases? Like in this section, we talked about the secondary biological bases, and we also talked about the data retrieval systems, and we studied the three database retrieval systems like sequence retrieval system, entries, dbget, and SRS is the most popular of the three. So thank you very much. We reached to an end. And I leave you with this cartoon to solve the query that whether the data is singular, but the facts which we get from it are singular or plural, like information is singular. So, I hope you enjoyed this talk, and if you have any queries, you can ask me anytime. Thank you very much for your patient listening. Thank you.